So in order to cluster these filtered data, what we're going to want to do is go up to our Partex Genomics Suite window, and we're going to click on Tools. The first choice is Discover, and what we want to do is pick the first choice there, Hierarchical Clustering. Click on that. This window will come up. Here it's basically just show it, giving you the parameters on how we want to group the genes and the columns. Also, how do we want to display the data? A lot of times you want to standardize the data, given that a lot of gene expression can be very, very different between different genes. We have already scaled this data, so we are not going to adjust the values at all. So I'm going to click None. Here we're asking how we want the clustering to go on both the samples or comparisons and the genes. Again, since we use scale data, and trust me on this, I've done this a couple times, we want to do rank Spearman dissimilarity as how we want to group it. Obviously, you can see there's a lot of choices there. We're going to do this for both. But given the fact that, again, we are using a rank system, I've tried other types of clustering methods. This seems to work the best and makes mathematical sense. We're going to leave the row method and column method. We're going to leave it average linkage. You can always put in how many clusters you potentially want in both the rows and the columns. And you could bootstrap the results. Here I'm just going to basically go with kind of the default except the changes that we made. So I'm going to hit OK. And so now what this will do is kick out our clustering on a new sheet. Now that we have our heat map up, we can format this any way we want. As you can see, we have the genes in the columns, and basically our comparisons are in the rows. What I want to do is switch that. I also want to change the colors. It's my opinion that green and red work for most people, but if you're colorblind, you're going to have a hard time basically getting anything out of these data. So what I'm going to do is, first off, is change the color. So what I'm going to do is, for genes that go down, I am going to turn that into blue, or actually blueberry. And for genes that go up, I'm going to make it different intensities of yellow. Okay, we are also going to want to transpose the rows and columns. So I'm going to switch those. I think everything's lined up here, and I'm going to hit Apply. So now we have the comparisons in the columns, and our genes are in our rows. As you can see, though, we're lacking some labeling. Actually, it's color-coded based on the geo number or the study it came from. What I want to do is put in our information on, say, our control tissue, what was our testing cancer that we used, what was the subtype of that cancer. To do that, what we're going to want to do is go to columns. And again, right now we have this new annotation. It's the geo. It's color-coded by the geo number. So I'm going to put in some new annotation. And I'll just do this for the first one, and then we'll use the magic of iMovie editing to put in the rest. So here I'm going to put in my control tissue. So I'm going to select control tissue. That comes up. The width in pixels is basically the width of the color bar. I've done this a couple times, so I'm going to put 22. I know that's a pretty good number. I want to show the label. And then for the label size, I'm going to put 20. Go down here. We can select the colors we want. Excellent survival. I don't want that red. I'm going to make that kind of green. So I'm going to put green here. Put OK. Normal, we're just going to put white. Normal adjacent, we'll make that gray or magnesium. Still gray. Sporadic, I'm going to make that strawberry. So I'm kind of going from blues to more red as far as the intensity of the more severe cancers. And then maybe type 1, we'll put that blue. Okay, and I'm just going to hit Apply. And now you can see that it is labeled. We can see the types of different comparisons, or at least the control tissue that we used. So now what I'm going to do, I'm going to basically do the same thing for the other cell types. So you should see an edit and the labeling will be there. And abracadabra, here is our labeled heat map. Our genes are clustered in the rows based on the parameters we clustered them with. Our comparisons are all in the columns, and again, they're labeled. Our first one is labeling by the geo number or the study. The second one is the control tissue we used in that comparison. The third column is the type of papillary renal cell carcinoma we used in the comparison, and then the last column, or the fourth column, is the subtype of that test, papillary renal cell carcinoma. I think this is fascinating. You can see a lot of agreement here. Again, blue is, if the more intense the blue color, the lower that gene was compared to the control, and the more intense the yellow color, the higher that gene expression was compared to the control. You can see that we're getting very distinct clustering here, that all of our comparisons involving cancer versus normal seem to be on the left, while our three comparisons looking at cancer versus cancer are on the right, and they look very different. And what's really nice is you can kind of use this to find specific groups. Also, I want to point out is even in the comparisons to cancer versus normal, we're seeing the type 2 kind of subcluster by itself. The nonspecific seems to be everywhere, which is probably made up of a mix of type 1 and type 2. I also want to point out here is we are getting a type 2 on this side, but this is the 2A. And if you remember back on my introductory lecture on the introduction of papillary renal cell carcinoma, the 2A, as far as survivability goes, behaves more like type 1 than type 2. And based on these gene expressions, I would say I would be tempted to group 2A with type 1. Again, you can see the patterns in here. You can see certain genes that seem to be higher in the type 2 versus the lower grade of that particular papillary renal cell carcinoma. So what we're going to do in the next series of videos is we're actually going to pull out some of these clusters, that these genes are more likely to interact together in some biological function given the fact that they behave similar across all these data sets. And you can see that with these genes here, and we can kind of select those here, that you are seeing an increase in expression compared to from the more severe to the less severe. And so we're going to use these two last columns to kind of figure out what's going on with this system and what are the different biological mechanisms that are probably playing a role in versus type 1 versus type 2. And so in the next videos, we will take these genes and export them into a text file and upload them into networking and bioinformatics software to pull out the biology. As always, thank you very much, and I will see you next week.